And welcome to Rex and Talking, the show a guy who's had MS since the turn of the century tried to keep up with speed. First order businesses at Seastead Retraction. I think those guys are okay. I mean, the girl, she's called Bitcoin Girl. I don't know her name. But anyway, she posted a movie about 6 days ago from Singapore where they were happily taking pictures of the lights. And I don't know, there's also this movie that shows the barnacles and coral and stuff that are going on the bar.
The day after it was floated, hundreds of fish appeared. They claimed Little Holes as their homes and defended it from other fish. This is how it looked after just 2 months. Barnacles are a nuisance for boats, but a blessing for seasteads. As mussels and sea plants grow, a new coral ecosystem will emerge. Since most of the surface ocean is a relatively lifeless desert, every seastead we float will increase the amount of life on the oceans.
I've started this blog a few times now, and I always get distracted or something. But this first thing is about lab-grown meat. I mean, I talked about lab-grown meat way back around blog 30. It was like 2 years ago. So really, the future has come to pass.
Would you eat a hamburger or a chicken nugget made of meat grown in a laboratory? Joshua Tetrick, co-founder and CEO of Just, is betting that you will.
In tomorrow's world, you can eat more meat, hopefully safer meat, even better tasting meat, without eating the animal.
Just, which is based in San Francisco, has been producing and selling non-animal versions of food like mayonnaise since 2013, and it's raised more than $310 million in venture capital. Tetrick and his team have created products like Just Mayo by identifying plant-based alternatives to common animal products like eggs.
We're 1 part pharma, 1 part biotech, and a whole bunch of regular food company. We build a discovery system that has the only patent in the world that utilizes machine learning for food ingredient discovery. And it helped us find this mung bean that when you put it in a pan, scrambles. And that process broken down means we source from over 51 countries in the world. We look at about 21 different molecular properties. We look to see how that bean functions. Does it bake a cake? Does it gel in a pan? And then we see if it makes ice cream or butter or scrambled eggs. Our process of finding these new tools is high tech, but what we find is not high tech at all. The mung bean has been in the world's food system for 4,400 years. I sure as hell didn't invent the mung bean.
Just is one of a handful of tech companies working to disrupt the meat production industry. While many of its competitors are pursuing better plant-based meat substitutes, Just is pushing ahead with so-called clean meat, or lab-grown animal tissue, that requires no farming, no feeding of livestock, and no slaughterhouses. Only a single sample from a single animal, duplicated endlessly.
We're on number 1. So where do you get your protein from? Nuts, beans, tofu seeds, quinoa, lentils, hemp and peas, oatmeal, spinach, and we know what's in it. I wish I could say the same for me. So's my boy and so's broccoli, but you can only still name me 1, 2. How do you afford to buy all that healthy food the exact same way you afforded to buy that iPhone, iPad, a brand new iMac, cars and clothes? You can't hide that your ego's bigger than your health. Try deny that and keep a straight face when you see yourself. 3, aren't we carnivores and supposed to eat meat? And that's why we have canine teeth to tear through the flesh and bones and meat. No, wrong again, they're there to break through nuts and seeds. And that's why we share the same teeth as a cow and a horse and all the monkeys. And the same size intestines. Told that when we're digesting, we take all the good from what we eat. They're not designed to digest meat. And carnivores have a lot more short because it's supposed to pass that speed. That's why we are left with disease such as cancer and diabetes. 4, so you're sure you're 100% a carnivore. Okay, next time that you go to the store, just grab some chicken, bacon or pork. Open it up and eat it raw with no seasoning or no sauce. No more knives and no more forks. And let's see who's hitting the floor, cause all carnivores have paws and claws. We have hands to pick fruit and veg off trees and plants, so please understand. If you put fruit and a rabbit in a baby's cart, it will play with the rabbit then eat the fruit. Do the same with the lion's cub, and what do you think that lion will do? 5, if they're alive, they'll overpopulate the whole earth. But you still proceed to inseminate them, no, so that they give birth. That's 56 billion animals each year that are sent to slaughter. That's 56 billion animals that you can feed and water. That's 8 times the fucking human race's population. Yet we can't stop hunger, poverty in every single nation. What? That shit don't sit with me, and it really, really gets to me when you hear them stupid fluoride hoes like, yeah, but what about they can know?
What we're going to try today are the first thin slices of steaks we have produced in a lab setting.
I think this is going to be the greatest revolution in the history of modern agriculture.
This lab-grown meat is not just a plant-based alternative like the Impossible Burger.
There's a technology that exists that allows us to, for all intents and purposes, have our bacon and eat it too, without any pigs being harmed.
Cultured meat, lab-grown meat, clean meat, whatever you want to call it, is identical to conventional meat at the cellular level, just grown in a lab, no slaughter involved. Its arrival could have massive implications for meat eaters, the US's $200 billion meat industry, and the environment. And it's happening at a pivotal time.
We can't handle more agriculture, and this growing demand is asking precisely that, and we're going to reach a breaking point.
This house was built in 24 hours. How? Thanks to modern technology, APISCOR 3D printing. The walls of this house were printed entirely on site. This is the world's first actually printed house.
I don't really have room to talk about 3D printing very much. It's one big topic. But really, let it be known that APISCOR, a Russian company, exists, and these houses are like 40 grand or quite cheap. But whatever.
Now, extinction is a different kind of death. It's bigger. We didn't really realize that until 1914, when the last passenger pigeon, a female named Martha, died at the Cincinnati Zoo. This had been the most abundant bird in the world. It had been in North America for 6 million years, so it wasn't here at all. Flocks that were a mile wide and 400 miles long used to darken the sun. Aldo Leopold said this was a biological storm, a feathered tempest. And indeed, it was a keystone species that enriched the entire eastern deciduous forest, from the Mississippi to the Atlantic, from Canada down to the Gulf. But it went from 5 billion birds to 0 in just a couple decades. What happened? Well, commercial hunting happened. These birds were hunted for meat that was sold by the ton. And it was easy to do because when those big flocks came down to the ground, they were so dense that hundreds of hunters and netters could show up and slaughter them by the tens of thousands. It was the cheapest source of protein in America. By the end of the century, there was nothing left but these beautiful skins in museum specimen drawers. There's an upside to the story. This made people realize that the same thing was about to happen to the American bison. And so these birds saved the buffaloes. But a lot of other animals weren't saved. The Carolina parakeet was a parrot that lit up backyards everywhere. It was hunted to death for its feathers. There's a bird that people liked on the East Coast called the heath hen. It was loved. They tried to protect it. It died anyway. A local newspaper spelled out, there's no survivor, there's no future, there's no life to be recreated in this form ever again. There's a sense of deep tragedy that goes with these things. And it happened to lots of birds that people loved. It happened to lots of mammals. Another keystone species is a famous animal called the European oryx. There's sort of a movie made about it recently. And the oryx was like the bison. This was an animal that basically kept the forest mixed with grasslands across the entire Europe and Asian continent, from Spain to Korea. The documentation on this animal goes back to the Lascaux cave paintings. The extinctions still go on. There's an ibex in Spain called Bucardo, went extinct in 2000. There was a marvelous animal, a marsupial wolf, called the thylacine in Tasmania, south of Australia, called the Tasmanian tiger. It was hunted until there were just a few left to die in zoos. A little bit of film was shot. Sorrow, anger, mourning. Don't mourn, organize. What if you found out that using the DNA in museum specimens and fossils, maybe up to 200,000 years old, could be used to bring species back? What would you do? Where would you start? Well, you start by finding out if the biotech is really there. I started with my wife, Ryan Phelan, who ran a biotech business called DNA Direct, and through her, one of her colleagues, George Church, one of the leading genetic engineers, who turned out to be also obsessed with passenger pigeons, and a lot of confidence that methodologies he was working on might actually do the deed. So he and Ryan organized and hosted a meeting at the Wyss Institute in Harvard, bringing together specialists in passenger pigeons, conservation ornithologists, bioethicists. And fortunately, passenger pigeon DNA had already been sequenced by a molecular biologist named Beth Shapiro. All she needed from those specimens at the Smithsonian was a little bit of toe pad tissue. Because down in there is what is called ancient DNA. It's DNA which is pretty badly fragmented, but with good techniques now, you can basically reassemble the whole genome. Then the question is, can you reassemble with that genome the whole bird? George Church thinks you can. So in his book, Regenesis, which I recommend, he has a chapter in the science of bringing back extinct species. And he has a machine called the Multiplex Automated Genome Engineering Machine. It's kind of like an evolution machine. You try combinations of genes that you write at the cell level and then in organs on a chip, and the ones that win that you can then put into a living organism, and it'll work. The precision of this, one of George's famous unreadable slides, nevertheless points out that there's a level of precision here right down to the individual base pair. Passenger pigeon has 1.3 billion base pairs in its genome. So what you're getting is the capability now of replacing one gene with another variation of that gene. It's called an allele. Well, that's what happens in normal hybridization anyway. So this is a form of synthetic hybridization of the genome of an extinct species with the genome of its closest living relative. Now, along the way, George points out that his technology, the technology of synthetic biology, is currently accelerating at 4 times the rate of Moore's law. It's been doing that since 2005, and it's likely to continue. Okay, the closest living relative of the passenger pigeon is the band-tailed pigeon. They're abundant. There's some around here. Genetically, the band-tailed pigeon already is mostly living passenger pigeon. There's just some bits that are band-tailed pigeon. If you replace those bits with passenger pigeon bits, you've got the extinct bird back cooing at you. Now, there's work to do. You have to figure out exactly what genes matter. So there's genes for the short tail in the band-tailed pigeon, genes for the long tail in the passenger pigeon, so on with the red eye, peach-colored breast, flocking, and so on. You add them all up, the result won't be perfect, but it should be perfect enough, because nature doesn't do perfect either. So this meeting in Boston led to three things. First off, Ryan and I decided to create a nonprofit called Revive and Restore that would push de-extinction generally and try to have it go in a responsible way. And we would push ahead with the passenger pigeon. Another direct result was a young grad student named Ben Novak, who'd been obsessed with passenger pigeons since he was 14 and had also learned how to work with ancient DNA, himself sequenced the passenger pigeon using money from his family and friends. We hired him full-time. Now, this photograph I took of him last year at the Smithsonian, he's looking down at Martha, the last passenger pigeon alive. So if he's successful, she won't be the last. The third result of the Boston meeting was the realization that there are scientists all over the world working on various forms of de-extinction, but they'd never met each other. And National Geographic got interested because National Geographic has the theory that the last century discovery was basically finding things, and in this century, discovery is basically making things. De-extinction falls in that category. So they hosted and funded this meeting of 35 scientists. They were conservation biologists, molecular biologists, basically meeting to see if they had work to do together. Some of these conservation biologists are pretty radical. There's 3 of them who are not just recreating ancient species, they're recreating extinct ecosystems in northern Siberia, in the Netherlands, and in Hawaii. Henry, from the Netherlands, with a Dutch last name I won't try to pronounce, is working on the aurochs. The aurochs is the ancestor of all domestic cattle. And so basically, its genome is alive, it's just unevenly distributed. So what they're doing is working with 7 breeds of primitive, hardy-looking cattle like that Marimana primitivo on the top there to rebuild over time with selective backbreeding the aurochs. Now, rewilding is moving faster in Korea than it is in America. And so the plan is, with these rewilded areas all over Europe, they will introduce the aurochs to do its old job, its old ecological role of clearing the somewhat barren closed canopy forest so that it has these biodiverse meadows in it. Another amazing story came from Alberto Fernandez Arias. Alberto worked with a bucardo in Spain. The last bucardo was a female named Celia, who was still alive, but then they captured her. They got a little bit of tissue from her ear. They cryopreserved it in liquid nitrogen, released her back to the wild, but a few months later, she was found dead under a fallen tree. They took the DNA from that ear. They planted it as a cloned egg in a goat. The pregnancy came to term, and a live baby bucardo was born. It was the first de-extinction in history. It was short-lived. Sometimes interspecies clones have respiration problems. This one had a malformed lung and died after 10 minutes. But Alberto is confident that cloning has moved along well since then, and this will move ahead, and eventually there will be a population of bucardos back in the mountains in northern Spain. Cryopreservation pioneer of great depth is Oliver Ryder. At the San Diego Zoo, his frozen zoo has collected the tissues from over 1,000 species over the last 35 years. Now, when it's frozen that deep, minus 196 degrees Celsius, the cells are intact and the DNA is intact. They're basically viable cells. So someone like Bob Lanza at Advanced Cell Technology took some of that tissue from an endangered animal called the Javan banteng, put it in a cow, the cow went to term, and what was born was a live, healthy baby Javan banteng who thrived and is still alive. The most exciting thing for Bob Lanza is the ability now to take any kind of cell with induced pluripotent stem cells, turn it into germ cells like sperm and eggs. So now we go to Mike McGrew, who's a scientist at Rosalind Institute in Scotland, and Mike's doing miracles with birds. So he'll take, say, falcon skin cells, fibroblasts, turn it into induced pluripotent stem cells. Since it's so pluripotent, it can become germplasm. He then has a way to put the germplasm into the embryo of a chicken egg so that that chicken will have basically the gonads of a falcon. You get a male and a female, each of those, and out of them comes falcons. Real falcons out of slightly doctored chickens. Van Novak was the youngest scientist at the meeting. He showed how all of this can be put together. The sequence of events. You'll put together the genomes of the band-tailed pigeon and the passenger pigeon. You'll take the techniques of George Church and get passenger pigeon DNA, the techniques of Robert Lanza and Michael McGrew, get that DNA into chicken gonads. Out of the chicken gonads, get passenger pigeon eggs and squabs. And now you're getting a population of passenger pigeons. It does raise the question of, they're not going to have passenger pigeon parents to teach them how to be a passenger pigeon. So what do you do about that? Well, birds are pretty hardwired as it happens, so most of that's already in their DNA. But to supplement it, part of Ben's idea is to use homing pigeons to help train the young passenger pigeons how to flock and how to find their way to their old nesting grounds and feeding grounds. There were some conservationists, really famous conservationists, like Stanley Temple, who's one of the founders of conservation biology, and Kate Jones from the IUCN, which does the Red List. They're excited about all this. They're also concerned that it might be competitive with the extremely important efforts to protect endangered species that are still alive, that haven't gone extinct yet. And so you want to work on protecting the animals out there. You want to work on getting the market for ivory in Asia down so you're not losing 25,000 elephants a year. But at the same time, conservation biologists are realizing that bad news bums people out. And so the Red List is really important to keep track of what's endangered and critically endangered and so on. But they're about to create what they call a Green List. And the Green List will have species that are doing fine, thank you. Species that were endangered, like the bald eagle, but they're much better off now, thanks to everybody's good work. And protected areas around the world that are very, very well managed. So basically, they're learning how to build on good news. And they see reviving extinct species is the kind of good news you might be able to build on.
Hello, Russell. He's a YouTube blogger. He's graduated business and stuff. He's interested in a lot of different things than me. But he's one of my favorite bloggers. And the other day, he posted a blog called Forged by Rivian. Rivian is an electric truck making company. They aren't even producing vehicles yet. Amazon bought $700 million of them too. So they've got $1 billion worth of investment. $1.25 billion of investments between Ford and Amazon. So that'll be big, because Ford is pretty loud electric vehicles. So Rivian, RIP, high-end, should be great. That would be awesome if next Ford F-150 had electric skateboard underneath it. That design the top. So the personal anecdote is that I sent my brother Hello Russ's blog, and I don't know what to do with it. But maybe that's why Ford bought or invested a huge